Hi, everyone. It's Dr. Levinsky. It's Rena. Vish, unfortunately, is having trouble introducing our speaker, so I think I'll just take over just for the sake of everyone's time. So thank you, everyone, for joining and for your patience. Today, we are so lucky to have Dr. Fissman coming to speak to us. If someone can update to the next slide, then I can actually give a good background history on our speaker. But in case people don't know, Dr. Fissman is a well-renowned epidemiologist working in Toronto. He has been instrumental in COVID epidemiological models at the beginning of the pandemic. Today, he's going to be speaking to us about the effect of climate change on infectious diseases. So I will pass the speaker to Dr. Fissman, and this might say some kind words at the end once his Zoom connection is better. Thank you, Dr. Fissman, so much for coming.
Thank you so much. Can everyone hear me okay?
Yes.
Oh, excellent. And I just need to be able to share my screen. I think that whoever is sharing these slides needs to stop sharing them. And while I'm waiting for that, thank you. I want to thank Dr. Levinsky and Dr. Joshi for this really kind invitation. It's marvelous to get a chance to speak about something other than COVID. Let's see if I can find my slides. Here we go. And go back to start at the beginning. So yes, really grateful to have a chance to talk about this. And apologies up front because it's 10 past 12. And as I was making these slides, I realized there is so much to say. I got a little bit carried away, and I think I probably have enough slides here to take us well through the afternoon and into the early evening, which I don't think folks will be hanging around for. So I'm going to try to do this at a fairly rapid clip. I may skip over a thing or two, but hoping that we can have time for some questions and some discussion. Before I start off, I have some disclosures. Both in terms of this being collaborative work, it's the work of many people, including Ashley Chute, Kevin Brown, Derek McFadden, and Amy Greer. We got funding for this work both from CIHR and the NIH when I was in the States. In terms of potential conflicts, I have served on advisory boards for a lot of vaccine companies. Pfizer, Merck, Seqirus, Sanofi, and AstraZeneca. I've been helping with an RSV model at Moderna as well, and I also teach epidemiology to Pfizer employees sometimes. I've also served as a paid legal expert during COVID for both the Ontario Nurses Association, Elementary Teachers Federation of Ontario. That's COVID, not climate change, but I have also served as a pro bono expert for eco-justice on climate change and infectious disease, and I've also provided consulting to a couple of financial companies, including Morgan Chase and Fairland Capital. So in terms of an outline, I thought we could start by just talking about climate change. One of the nice things, it's not great to be talking about climate change at a point in time when, it seems pretty apparent to any observer that the climate has changed profoundly. If you have the ability to think back more than 10 or 15 years, we've just sort of come through a non-winter in Toronto. So at least we won't have to spend time arguing about whether or not climate change is happening, because that did tend to hang things up 10 or 15 years ago. But we'll talk a bit about basic concepts around climate change, and then I'd like to pivot to talking about ecology and infectious diseases as ecological entities and what that means. And then a couple of very brief slides just on some of the research challenges in this area, and also some of the implications of climate change for both public health surveillance and for clinical practice. So without further ado, here's an old slide. And again, a lot of this has been known for a long time. It's just we've had this sort of merchants of doubt thing going on with folks kind of ragging the puck and creating a lot of uncertainty. But we've known that the planet is warming. Not only is it warming, but it's warming at an accelerating rate. This is from the Intergovernmental Panel on Climate Change panel assessment from 2007, and you can clearly see what's happening in terms of the trend lines. They're doing trend lines over shorter and shorter periods of time, this graph on the right, and what you can see is the slope of these lines becomes steeper and steeper the shorter the period that they use for this regression model, and that simply tells you that climate change is speeding up. We think it's mostly attributable to our actions as humans. When folks have been modeling climate change, the pink shaded areas here represent climate change models that incorporate greenhouse gases, mostly carbon dioxide and methane. The black line is the actual data, and then the blue line is the blue shaded area is trying to run those same models for global temperatures, land temperatures, and sea temperatures without greenhouse gases. And what you see is that you can't kind of create these trends if you model what's going on, but you leave greenhouse gases behind. So we do believe that this is attributable to human action, human generation of greenhouse gases. The projections, again, this is an old slide, but it's held up well. The IPCC projections from 2007 were that we'll experience in North America increased temperatures, increased rainfall. We've lived that. Increased drought and wildfire. We lived that last summer, and increased frequency of extreme weather events. One of the disconcerting things, though, is for all that those projections and models worked very well, this past year they actually started to fall apart. So what you can see here, this is from Copernicus in the European Union, scientific body in the European Union, looking at global surface temperatures. It's sort of a spaghetti plot. The blue on the bottom is earlier time periods, 1940s, 1950s, 1960s. And what you can see is the global mean temperatures get higher and higher and higher, as denoted by the increasingly orangish colors. And then on the top of all that, you've got 2023, last year, where it looks like we've actually flipped over to a different kind of system. It's just a complete departure from what was happening before. We also see that with sea ice. The blue curve is last year's Antarctic sea ice cover, which is just completely departed from previous curves in terms of being at a new minimum. Gavin Schmidt, who used to run NOAA, the government agency on atmospheric science and climate in the United States, penned an editorial in Nature last month, I guess March 19th, I think, where he pointed out that our models are now failing, that we now have warming happening at an extreme rate that wouldn't have been predicted by models. And that's disconcerting for people in this field for a couple of reasons. One is that when we don't understand systems, the same applies to infectious disease models. When we don't understand systems, it becomes very hard to predict and hard to control. We don't know what's coming next. And we're also aware that there are tipping points baked into climate systems where we can get positive feedbacks. So just as with an epidemic, the more cases you have, the more cases you get, because the more people are doing infecting. With climate change, when we lose, that shouldn't say Arctic, it should be polar ice cover. When we have forest fires created by, generated by climate change, when we have methane released from permafrost, all of those things are both products of climate change, but they also accelerate climate change. Forest fires obviously release a ton of CO2. When we lose polar ice cover, we go from white poles that reflect heat to dark poles, and that contributes to ocean warming. And then, of course, when we get permafrost melting, we have a lot of carbon locked into permafrost over vast areas. And when we get permafrost melting, we get that carbon released in the form of methane, and that further accelerates climate change. So we may have hit some kind of tipping point, or maybe hitting that at the moment. We know, this is another old slide. All the green bars, this is from a reinsurance company. A lot of the really good data on the effects of climate change actually come from insurers. There's a company called Munich Re, which is a reinsurance company, which is an insurance company for insurance companies. And they have a magnificent database on climate change effects, because of course they have a vested financial interest in what's coming down the pike. This is to 2009, and what you can see is all the non-blue and non-red, all the non-red bar components of these bars in terms of major natural catastrophes in the US. They defined this as more than $1 billion in payout or more than 50 people dead. All of the non-red aspects of this, which is storms, floods, winter storms. I'm not sure what other is, but they sort of were increasing exponentially. And this is, that's a 15-year-old slide. But what you see if you go on Munich Re's website now is they've got a lot of great data on what's happening with natural disasters and climate change. We have convective storms markedly increasing. This is US data that they had a 90% increase in economic losses year on year, 2022 to 2023. A lot of that is climate change related, also relates to population growth. They've got a similar figure showing that wildfires, this is California data, have taken off since the year 2000, as models would have predicted. And we even have projections of increases in events that are very interesting to insurers, like extreme hailstorms, which do a lot of damage to property. And extreme hailstorms here are defined as hailstorms with hailstone size greater than 5 centimeters. And that sounds strange. You talk about a warming world, and yet we have more dramatic ice storms. And this simply relates to the amount of energy, thermal energy in the atmosphere. Hailstorms, massive hailstorms, are a product of the strength of updrafts within these convective storms. So if you get small granules of ice pushed way high up into where you have liquid water that's super cooled, that's where you get the recipe for massive hailstones. Of course, this is a medical talk, so just to move away from the insurance industry, these changes obviously have implications for human health. We can have heat-related mortality events. You may have taken part in care during these. We have respiratory health issues in terms of fires generating particulates and other pollutants. We know PM 2.5s in and of themselves can trigger myocardial infarct and cardiac arrhythmias. We can have injuries due to hurricanes, tornadoes, and fires. And then we can have secondary health effects of disrupted economies and displacement of populations via coastal flooding and desertification. Just to be a little bit more on the upside, more on the optimistic side, there's a marvelous organization called the Climate Health Alliance in Australia that talks about the fact that as healthcare providers, as physicians, as people who have a lot of cred with the public, climate change really is a health issue, and climate change mitigation can be a health issue. And they've coined this term, the health dividends of climate change mitigation. That if we move more away from passive transportation with carbon fuels, if we move more towards active transportation, if we move more towards wind and solar rather than burning fossil fuels for energy generation and so forth, the knock-on effects of that on chronic disease risk and on respiratory disease risk and cancer associated with burning fuels potentially provides a health dividend. So we do well for the climate, but we also do well for population health. But I'm here really to talk about infectious diseases. So what's an ecosystem? Climate change has important effects on ecosystems. Ecosystems are just complex biological systems that have both living and non-living components. So when the climate changes, we have changing temperatures, changing water ability, changing ocean pH via CO2, changing frequency of extreme events, fires, floods, extreme rainfall, and so forth. So the physical components of ecosystems are changing, and that stresses the biological, the living components of ecosystems. And we can expect both agents of disease, microbes, and also their vectors and reservoirs are components of ecosystems. So when we stress them by changing the physical component of ecosystems, we expect that the living components of ecosystems also adapt, change, or are stressed in some way. And we would expect to see changes in incidence and distribution of infectious diseases as a result. We might also see some more complex causal pathways, for example, enhanced infectious disease transmission due to displacement of populations, as mentioned on the previous slide. So if we have refugee crises, we know social disruption, war, people living in crowded conditions in camps are all great for transmission of communicable disease. So to be more specific, in terms of potential impacts on infectious diseases, I'm going to start off by talking about vector-borne diseases because that's sort of the easiest one to intuit. If we have changing ecosystems, we may change the ranges of amplifying hosts, and we may also change the time intervals involved in the amplification of those diseases. When we have diseases with environmental reservoirs, which I'll also try to talk about in water and soil, these aren't communicable diseases, but these are diseases that we get from the physical environment around us. That may have effects on pathogens like Legionella, gram-negative rods that live in surface waters and soil, impacts on our water sources. We might even experience inoculation via extreme weather events, and I'll show you a brief reference to that in terms of a disease called melioidosis that's been associated with monsoons in the Pacific area. And we could also potentially see perturbations of communicable diseases. These are diseases transmitted from person to person. So none of this is really that new. A lot of what I'm going to cover in my talk, we didn't have some of the more recent data, but in general terms, we actually did talk about this in the CMAJ in 2008, and this was a review article that was about 2 years coming. So we truly have known a lot of this for about 20 years. So in terms of talking about vector-borne diseases, I'm just going to give you, let's imagine this is West Nile infection or similar mosquito-borne disease. How might climate change increase the burden of that disease? Well, let's say in our current scenario, or in our scenario 20 years ago now, we have the following ecology for mosquitoes. So just look at where it says current scenario. Mosquitoes aren't flying around biting people during a cold winter. They're overwintering in catch basins and sewers. A lot of them don't survive the winter. They get activated as temperatures warm. Once they're activated, they can lay eggs. For a disease like West Nile, we have transovarial passage of the virus, so they lay infected baby mosquitoes. It takes time for those larvae to develop into biting mosquitoes. And then for West Nile, we have this amplification process because mosquitoes that spread West Nile tend to feed preferentially on birds. But you have this mosquito bird amplification cycle that takes a little while until you get so much West Nile in mosquitoes that bridge vectors, mosquitoes that will happily bite either a bird or a human, start to get infected, and then those folks, those mosquitoes generate this crossover from birds to humans, and then in October things freeze up, and the mosquitoes stop biting. So that's our historical ecology of these diseases. Under a warming scenario, what you have is spring temperatures onset earlier, or maybe even we don't even have a winter, as this past year. We have eggs laid earlier, but larval development's also temperature sensitive. So we get our mosquito populations maturing earlier. We get a more rapid onset of the amplification cycle, which means we get a longer, we get earlier crossover to humans, and we get a longer time before the vectors die off in the winter. And I should have stretched this out at the right side of the screen as well, because winter comes later. So that's predicted to lead to a longer duration of exposure for human populations and a higher burden of disease. We can also expect, and we're going to circle back to this, we can also expect to see vector ranges changing. This is some old modeling, again, about 20 years ago, from my colleague Nick Ogden, who's now at Public Health Agency of Canada. When I was in medical school, I graduated in 1994, and at that time, the belief was that the only place in Ontario that you could get Lyme disease was Point Pelee Provincial Park, which is about at the latitude of Northern California. And what we've seen is this rapid northern extension of black-legged tick ranges, simply because the climatic conditions at further and further north latitudes allow these black-legged ticks to actually complete their life cycles. And we'll circle back to that in a moment. So we can see increased burden because of the changing ecology of the disease. We can see increased burden because of the changing range of the disease. And the changing range of the disease for vector-borne diseases can also involve altitude because it's colder higher up. And I'll skip through this slide pretty quick, but this is by an ecologist at University of Michigan named Mercedes Pascual, where they actually had some data. I'll just direct you to the top and bottom left-hand panels of this graph. They had case data on malaria from Ethiopia and Colombia, and these are plots of the average altitude at which cases lived, because they're in mountainous areas. They have the home address of malaria cases, and they're able to show, you can see the orange curve is shifted over relative to the black curve, that malaria is moving higher and higher up these mountains. And of course, historically, folks who live in malarious areas have tended to build at altitude for precisely this reason. If you get up higher, I think even the Pope's summer residence, Castel Gandolfo in Italy, was built at altitude to get the Pope away from Rome during the malaria season. But that is no longer, protective. We have these diseases actually moving up at altitude. So we can get changing ecology, we can get changing range, and then we can also get changes in epidemiology associated with acute disruptions in weather, extreme weather events, changing temperature, changing rainfall. I'll skip through this pretty quick. This is some work that we did, again, probably 15 years ago. Jonathan Soverow, I thought, would get into infectious diseases. He was a medical student at the time. I believe he's gone off and become a critical care doctor. But he did this lovely study where he got some great data on West Nile cases from the US CDC. There's the cases in terms of where they fall seasonally over a number of years. And Jonathan did what's called a case crossover study, which I can sort of unpack later if folks are interested, but looking at different environmental effects and what they did to West Nile risk. And what you can see in the bottom left panel, this is panel C, is when it rains really hard a week or two prior, that really makes risk of West Nile go up. I mean, you'll all know mosquitoes have pretty short generation times. When there's a lot of rain, there's a lot of habitat for them to lay eggs in and breed in. Whereas temperature effects are seen at both short legs, this is panel A, top left, short legs, and at longer legs. And that's one of the interesting things when we get into climate and infectious diseases, is the same exposure at different legs can impact epidemiology in different ways. So at 1 to 7 days, what we're probably seeing there is mosquitoes bite more frequently when it's warmer. They're very temperature sensitive. Out at 22 to 28 days, we're probably not seeing biting. What we're probably seeing is more stuff related to mosquito survival and reproduction in the environment. So you have the same exposure impacting risk at multiple legs. To circle back to Lyme disease, just a reminder in terms of the epidemiology of Lyme disease, we're collateral damage with Lyme disease. The important stuff happens with black-legged ticks and white-footed mice mostly. But a gravid tick lays eggs that turn into larvae, and these larvae need to, it's sort of like their 10-year plan, except it's only 1 year. They need to be able to mature before wintertime comes again, mature, lay their eggs, and then they can die having accomplished their life's work. But in order for their progeny to be successful progeny, they need a blood feed for every molt. So what you need to see with what you see with climate change is that we've changed the amount of territory where black-legged ticks can actually achieve 3 feeds before the winter freeze up, before they die. And as mentioned, here's Nick Ogden's model again, suggesting that based on that, based on the amount of territory where black-legged ticks can actually achieve their life cycles, we expect to see Lyme disease marching north in Canada over time. But you can see that if any of you are aware of the epidemiology of Lyme in Ontario, the 2020s actually looks a little bit stingy in terms of how far north this has gone. And indeed, this is from a number of years ago from Mike Parkins at University of Calgary, where they actually diagnosed a case of human granulocytic anaplasmosis. Some of you may know it as ehrlichiosis, which is also spread by black-legged ticks in Calgary in a guy who had never traveled. So, the actual ecology of these diseases may be outpacing our models somewhat. We did some work a number of years ago where we looked at US Lyme disease. We used US state Lyme disease simply because we could get the data and we couldn't get provincial data for Canada. And you see there's this upward trend over time in case numbers, and that could be due to a number of different things. But what we did is we broke it down by latitude, and what we saw is that Lyme disease is actually increasing in northern states, seems to be not changing in these yellow states, and then in some southern states actually seems to be decreasing rather than increasing. And what we did is this is what's called a meta-regression plot. We're looking at latitude and we're looking at rate of increase. And what you can see is whether we look at northern states, southern states, or all states, what we see is we see the rapidity of increase of Lyme disease cases is related to latitude. Which is the effect that you would expect to see if this was driven by warming, by climate change. You have more warm weather further north than you did before. In terms of why things might actually be going down in the south, there were a couple of hypotheses for that, which is why we stratified by northern and southern states. One hypothesis was that the US CDC was really encouraging southern physicians to do testing for Lyme disease rather than diagnose it clinically, because there's something called STARI. Some of you may be familiar with southern tick-associated rash illness that looks like Lyme, but is not Lyme. Another hypothesis is that you actually have northern range expansion of lizards in the United States, which are dead-end hosts for Borrelia burgdorferi. So that the black-legged ticks can substitute lizards for mice, and whereas mice are competent reservoirs for Lyme disease, lizards are not. I'm not sure what the latest is on that debate. So one of the problems, and I'll circle back to this at the end, one of the problems with studying climate change and infectious diseases is that we only have one planet. So we don't have a control planet without climate change where we can compare and say, oh, this is definitely due to climate change. And we were inspired a few years ago by some work by a guy named Solomon Sang, who was using the El Nino Southern Oscillation. We're in an El Nino year this year. It's basically an irregular periodic warming of the Pacific Ocean that really makes weather go crazy, particularly in the Western Pacific area. Sorry, that would be the Eastern Pacific to us, sort of the coast of South America, coast of California. And Solomon wanted to study, what's climate change going to do to conflicts and wars? Because there is the belief that as water gets scarce, as crops fail, as people get hungry, as people are on the move, that's going to spark conflict. And so he used El Nino as kind of an experimental system to look at the relationship between conflict and El Nino. And he did find that in areas where the weather's strongly affected by El Nino, you do seem to see this association with conflict, and you didn't see it in areas where you don't expect the weather to be as much affected. So we took that a couple of years ago and used it to look at infectious diseases in the United States using something called National Healthcare Discharge Survey. And what we found is we found it to be quite useful in terms of looking at vector-borne diseases. We wound up aggregating up. You see an El Nino effect with about a 1-year lag. So when it's a very El Nino-y, hot, wet year in the Western United States, a year later, what you seem to see is more vector-borne disease risk. And this can be Lyme disease. It can also be mosquito-borne disease. And there are probably a bunch of different mechanisms underlying this. As I say, the legs, when you're studying this, can be really tricky. One of the issues with, for example, Lyme disease can be that you actually have warm, wet years result in more vegetation, which results in more rodents, which results in more rodents having birth. And those rodents then give you a more robust amplification cycle for a disease like Lyme disease the next year. So the lags can be tricky, but we saw this really big increase in risk with El Nino at a 1-year lag. Now, the last thing I'd like to talk about is actually emerging infectious diseases. So either we've just come through one of those in terms of COVID. I guess we still have it, but it was a pretty impressive emergence. So emerging infectious diseases have this very woolly definition. They're diseases that either are newly described or they're diseases that are changing in their epidemiology or distribution. So non-specific as to be pretty useless. But anyway, be that as it may, when we think about emergence, what I tell students is new infectious diseases, we have to think about them along two dimensions. One is it's almost like growing a garden. It's seed and soil. So we need a novel pathogen. In COVID, we had that. Don't need to get into where it came from, but we had a novel pathogen, but we also need fertile soil. And what we have seen with some infectious diseases that haven't taken off, and touch wood, so far this applies to H5N1 bird flu, is for it to become a communicable disease, it actually needs the pathogen characteristics and the population characteristics need to be such that each old case can make more than one new case before it goes away, and then the disease will become established. And we quantify that in epidemiology as the reproduction number. When we have reproduction numbers less than one, we can see small clusters, as we have seen with H5N1, small family clusters. Sometimes we've had, usually a kid who has H5N1 and then a parent caregiver gets it, and then you have a cluster of size 2, but it's a dead-end cluster. This was described a number of years ago in Nature, in terms of zoonotic crossover from animals to humans. And we can think about this in terms of reproduction numbers. If we have something like rabies, we may have only animal to humans, no onward transmission from humans. If we have something like H5N1 right now, we may even see an occasional secondary infection, but we don't see sustained chains of transmission. The worry is of course that we have a pathogen with sustained chains of person-to-person transmission, and then the pathogen becomes established as an endemic source of disease. So there's been a lot of focus. We're in an era, we've forgotten about it, I think, a little bit because of the pandemic, but we really are in a remarkable era of emerging vector-borne diseases. In terms of Western Hemisphere emergence, all of these different viruses are either newly recognized, like they'd never been seen before, they'd never occurred in the Western Hemisphere before. Since 2000, we've got chikungunya, Zika, Mayara, Oropouche. I don't know what Itaqui virus is. Heartland virus, newly identified in, I think, Indiana. Powassan seems to be expanding its range. West Nile was never seen in the Western Hemisphere. And we've also got a marked increase, apparently, in dengue, although some of that may also be due to diagnosis. But I mean, when you look at the literature around Zika from, I think it's 2014, 2015, there's this tremendous focus on who brought Zika to South America. And my contention is Zika's probably been introduced into South America innumerable times over the last number of centuries. Tens of millions of people were transported over a period of about 300 years from sub-Saharan Africa to Brazil in particular, as part of the slave trade. And what we know from phylogenetic data is that falciparum malaria in South America, which involves Anopheline mosquitoes, that those introductions phylogenetically came with the slave trade, just in terms of genetic clock analysis. We know that yellow fever, which is endemic in South America, and shares a vector with Zika and chikungunya in terms of sharing an Aedes vector. We know that that's likely to have also been introduced between the 1500s and the 1700s, again, via the slave trade. And when we look at where Zika and chikungunya are endemic in Africa, you've actually got this little light green slice that I think is probably due to non-reporting for chikungunya. And you look at where where the epicenter of the transatlantic slave trade was, which is Ouida and Benin, was famously the disembarkation point for these slave ships. It's right in the middle of Zika and chikungunya, the Zika and chikungunya zone. So the idea that these were just suddenly introduced when Brazil hosted the World Cup doesn't make sense to me. What we do know, this is a reproduction number. Sorry, it's a complex formula. Don't worry about it. For the reproduction number for malaria. And what you see there is this little squared term in that model. And that squared term is biting rate. So there's this nonlinear relationship for the reproduction number of malaria with biting rate of mosquitoes. Why would that be? Well, when mosquitoes bite more, mosquitoes infect more humans, but the mosquitoes themselves also are more likely to get infected by humans. So you actually have this exponential increase in transmissibility as biting rate increases. What increases biting rate? Well, temperature increases biting rate. And if we do a thought experiment for a disease like let's say, whoops-a-daisy, like Zika or chikungunya, where at baseline we have a reproduction number that's just below 1, and then we slightly increase biting rate by increasing temperature, what we're going to get is we're going to get an increase in reproduction number. We're going to get a reproduction number that goes above 1. And that means that the disease can cause an epidemic, as both Zika and chikungunya did, and then become endemic, because population immunity is going to push that reproduction number back down to 1 by depleting potential hosts, but it remains endemic. And I think that's probably what we've seen with the emergence of some of these diseases. Now, the last thing I want to throw out to you, which is probably a little bit funny and contentious, but actually has been incorporated into food safety practice, is the idea that we think of vector-borne diseases as just being diseases of biting insects. In fact, if you think about the creatures in the environment that have a lot of taxus for both feces and for food, you would probably realize pretty quickly that houseflies really are the agents in the environment that will seek out a plate of food and will also seek out a poop. And there was a realization a number of years ago that flies were probably driving a lot of campylobacteriosis, a lot of the seasonality of campylobacteriosis. That's been responded to by actually putting fly nets up in abattoirs to protect chicken carcasses, with a resulting reduction in campylobacteriosis incidence. But this is some work for Melanie Cousins, who was a grad student at University of Guelph a couple of years ago, looking at climate change effects, because the generation times don't just shrink for mosquitoes, they also shrink for flies under climate change scenarios. So we could even think about climate change driving changes in foodborne disease epidemiology if we treat them as vector-borne diseases. Now, the last thing I'd like to talk about in terms of vector-borne diseases, and I'm going to zoom real quick through some stuff on environmentally abundant diseases, is the role of poverty, because risk isn't distributed equally. This is one of my favorite papers ever. It's by someone named J.M. Brunkard, who I don't know, from the US CDC, who took a look at dengue seroprevalence, again, another vector-borne disease spread by Aedes mosquitoes, in the city of Brownsville/Matamoros, Mexico. So Brownsville, Matamoros is actually a single city, but it has an international border down the middle of it. It's kind of a massive urban area that crosses the Texas-Mexico border. And what they did is they did a seroprevalence study looking across that border at what the differences were between Americans and Mexicans in terms of dengue seroprevalence, because, a border wall or a border fence isn't a deterrent to mosquitoes. So you should have a homogeneous population of mosquitoes, you have humans on both sides, they should have equivalent seroprevalence if risk is equal, and yet they do not. So the Mexicans had much higher dengue seroprevalence than the Americans. And when they dug down in terms of what some of the predictors were of higher seroprevalence, they found that they were mostly economic. So in terms of water, the Texans get their water through pipes. The Mexicans were more likely to get water from rooftop tanks. The Texans stayed cool by opening, by using air conditioning. The Mexicans were more likely to have open windows. When they looked at potholes and street drainage, they found that potholes per square mile or square kilometer was associated with dengue risk, which makes sense because potholes fill up with rain and become breeding habitat. So risk isn't distributed equally when we look at vector-borne disease risk. It's very tied up in economics. And I see we're already at, I'm going to ask my hosts, we're already at 10 to the hour. So how would you like me to proceed? Or how would the audience like to proceed? What I was going to talk about next was infectious diseases with environmental reservoirs, so water, soil. But we can also break and just do questions until 1. What do you think? Okay, not sure if I'm not getting any feedback. So I'm just going to go for a little bit longer and talk about infectious diseases with environmental reservoirs. Oh, there's someone said something. Keep going. Okay. Okay. And you pull the plug on me when you're ready to pull the plug on me. Okay. So whoopsie-daisy. There we go. So in terms of infectious diseases with environmental reservoirs, let's start off by talking about Legionella, which again, just like talking about vector-borne diseases, this is sort of an easy entree into the topic. So we know that Legionella incidence is going up and up and up. People argue about why that is. Is that more diagnosis? Is that more disease? Is that just an aging population, more immune suppressed individuals in the population? So we haven't nailed that down. However, we do know that Legionellosis is extremely climate sensitive. And this was some work we did years ago. My colleague Suette Lim at the Philadelphia Department of Public Health approached me when I was at Drexel and said, we should look at, it's been weirdly rainy and we've had a lot of Legionella. And we had a collaborative study involving the 5 counties, just like the GTA involves five big municipalities. Philadelphia is very similar. It's a 5-county area for Greater Philadelphia, and everyone was very cooperative in terms of sharing case data. And we, of course, had our weather data, and we were able to show that wet weather is associated with spiking risk of Legionellosis. So this is, I think, the paper title I'm proudest of all time is it's not the heat, it's the humidity, because we know that Legionella is a summertime disease, but what seems to be the driver is actually the wet weather, rainfall and humidity. Something that's really lovely is I've become connected with she's not a graduate student anymore, just graduated, Kelsey Castle at Yale University, who's just moved to Merck. Kelsey, for her PhD, actually redid our study, but did it with a spatial component in Connecticut. She was at Yale. And what she found was that there are dramatically different incidences of Legionella in Connecticut, where a couple of the river basins, these areas in blue, are really their hotspots for Legionella, and the rest of the state has risk that's much lower. And what she found was that within those watersheds, these areas around rivers, well water, if you're a well water recipient in those areas, that seems to be very important in driving Legionella risk. And we know Legionella is environmentally abundant. But Kelsey also did a bunch of analyses that repeated the stuff we had done in Philadelphia, and again found this strong association with humidity, strong association with rainfall, a strong association with stream flow. All of these are climate sensitive exposures. So what we know about Legionella is that it has something to do with the environment. We know it's environmentally abundant. We know it's abundant in surface waters. Does rainfall create aerosols, out in outdoor spaces when we're walking around? Does rainfall drive micronutrients into water processing plants and result in proliferation of Legionella in water in source waters, we still don't know. But the idea that Legionella is possibly ingested and aspirated, or is inhaled, when you're in the shower or what have you, those are some of the hypotheses about how it gets from aerosols that are around you into your lungs. But it does seem to be climate sensitive. Here's another one that probably doesn't surprise you. This is some old work that we did during the Haiti. Well, I mean, I was going to say during the crisis in Haiti. Haiti's in crisis. So I almost feel like this is the earthquake and cholera epidemic in Haiti were the beginning of the great unraveling of Haiti. But we did some work on folks who post-earthquake in 2010 were in refugee camps, and we were able to show a strong relationship with rainfall. Presumably, this is odds ratio on the y-axis and days prior to case occurrence. Looking at rainfall, there's this sharp spike in rainfall, in risk with rainfall. And you could imagine, oh, of course, well, you don't have good sewage disposal. People have water sources that aren't protected. There's a big rain. It's just going to wash fecal material into water sources and people are going to get sick. Oh, well, that's a disease of poverty, and that's in Haiti where they had no infrastructure. But you look at our waterborne, at the Walkerton water disaster in Ontario, Canada in 2004, you have exactly the same effect where these lines are extreme rainfall events. A lot of people forget about Walkerton that the disaster itself was preceded by once in 100 year at that time rainfall events, which probably washed a lot of cow manure into well sources. This was in an area that had a lot of cattle farming and where a lot of people got their water from wells. So extreme rainfall events can cause contamination of drinking water sources. So rainfall is one component of this. Here's some other work by some students, Rebecca Ling and Chloe Logar Henderson, where they looked at non-cholera vibriosis. So we talked about cholera, but there are, of course, Vibrio vulnificus and friends that can cause both bacteremia, can cause sepsis with sort of bullous skin lesions, and can also cause foodborne enteric illness. And these are also diseases where incidence has been rising. These are bacteria that are abundant in brackish waters in areas where salt and fresh water mix. Similarly, they've been going up and up. Again, we're using US data because we could get a hold of it. And whoops-a-daisy, I thought I had a little animated box there. What you can see is Rebecca and Chloe looked at an El Nino effect. And what you have is per increase in the El Nino index, so there's a 3-level index, you get about 14-15% increase in your Vibrio case count. You also get much as we did with Lyme disease, you also see this dose-response relationship between the increase in rate and how far north places are. Again, implying that this may have something to do with oceans getting warmer at further north latitudes. Doesn't prove it, but it would be consistent with that. I think I just have time to talk a little bit about gram-negative bacteremia and antibiotic-resistant organisms, thanks to Len Mermel, who's a colleague at Brown. We did a study a number of years ago, and Len's a great networker, so he got microbiologists from all these different cities to give us bacteremia data. And what we found is that gram-negative bacteremia is seasonal. We weren't the first ones to show it, but you sure see it. This is the solid curve is the average seasonality for northern latitudes. The southern latitudes are the dashed curve. So and when we see things that are profoundly seasonal, that may imply environmental influence. And what we saw is that the closer you are to the equator, the greater the fraction of your bacteremia is gram-negative. So this sort of implies, gram-negatives, a lot of gram-negative rods are environmentally abundant. They're in sewage, they live in surface waters, they're in soil. And there may be something about warmer temperatures that increase human risk for gram-negative bacteremia. What we know from Derek McFadden, who's a former trainee at U of T, who's now a professor at University of Ottawa, is that not only do we get increased risk of gram-negative bacteremia, but we also see an association between resistance in gram-negative pathogens and temperature. So Derek sort of did this lovely heat map of he has a normalized resistance index, and you can see southern US versus northern US. There's kind of this south to north gradient, and he found that it was class specific in terms of it being mostly fluoroquinolones and beta-lactams. Now, how would that work? Well, again, these bugs are environmentally abundant, and um, they are going to be their replication and their evolution is going to be temperature sensitive. So if we have bacteria, let's say in a pond, and we also have sewage contamination in that pond, and we also have antibiotics in that pond because we do have drugs and metabolites of drugs that get out into the environment. What we're going to have at warmer temperatures is both more rapid natural selection and also more rapid lateral gene transfer, which seems to be an important piece of the puzzle for antibiotic resistance moving from bug to bug. Yeah, I'm going to just skip through this in terms of extreme weather events, environmentally abundant pathogens, because I want to get this in before I stop. We have the same model. This is from Arturo Casadevall in New York, in terms of whether thermotolerant Candida auris may have emerged via the same route. Candida auris has been an environmentally abundant yeast for a long time, but it's never really made the jump to humans until the last couple of years. Arturo's contention is that as we have warming wetlands, which is where Candida auris has historically been found, you've created a thermotolerant yeast that can now replicate in a human bloodstream, whereas previously with colder surface waters it couldn't. That's a hypothesis. There's not hard data to back that up. And we've got a bunch of other data on changing fungal dynamics I don't think we have time for. In terms of zoonotic disease, don't have time to discuss it at length. I think these slides are being shared. If anyone wants to follow up on some good references, there's a couple of references at the bottom here. But again, zoonotic diseases are in Canada are disproportionately a threat in northern Canada and disproportionately impact northern First Nations communities. So when we think about impacts on fox populations, impacts on caribou populations, life cycles of things like echinococcus, where we already have First Nations people up north being at disproportionate risk. When we think about diseases like tularemia that seem to be emerging as Arctic diseases, when we think about let's see, so sorry, tularemia, echinococcus, and when we think about shifting predator populations, as we're seeing with foxes, that may impact rabies, a lot of that is going to be disproportionately felt in the north. I had a couple of slides to sum up. I'm not sure if I should what I should do here, if I should leave it at that, or maybe I'll just try quickly to sum up. You don't want to know about the research challenges. Someone can ask what the research challenges are if we get that in questions. So in terms of surveillance and clinical practice, I want to end with this. So surveillance is the cornerstone of public health. You can't manage what you can't see. If we don't measure it, we're flying blind. Lyme disease, we had a tautology in Canada until 2009 that we didn't have Lyme here. So if you got a disease that looked like Lyme and you were in Canada, it couldn't possibly be Lyme. Now Lyme is nationally notifiable, and we're starting to understand the epidemiology in Canada, but we also have other diseases with climate links, including echinococcosis, blastomycosis. Blastomycosis, Canada is the world's hotspot for blasto, that are not nationally notifiable, though both of those, I will say, have been reportable in Ontario for the last couple of years. How does that translate to clinical practice? Well, one of our heuristics when we're diagnosing is, is this a disease that occurs here? And if you don't measure it and you don't report it, it may very well occur here, but you would never know it. This is just some clinical decision-making literature. We're often dependent on pretest probabilities. If we don't do surveillance, we don't know what the pretest probability of a disease is. Indeed, we might not even know that it's here. So with that, I will end. And sorry, as I said at the beginning, I did make too many slides, but I was kind of excited about all this stuff, and I hope that was useful to you.
I just wanted to thank Dr. Fishman on behalf of the entire Department of Medicine. That was a very engaging discussion. I think we're all going to be extra careful on how we impact climate change in our day-to-day activities. And I think already the ID specialists at the hospital are well aware of the increase in Lyme. And I think Ontario has done a great job of trying to do extra surveillance so we can actually identify the problem. So I just want to thank you so much for taking the time to create this talk individually for us, which was fantastic. And if anyone has any quick questions, we'll be able to take them for about a minute, maybe just one question if you want to raise your hand, just because we're really over time and I know people have to get back to work.
Yeah.
So I don't see any hands raised.
Okay.
Dr. Fishman has been kind enough to give us his email if you want to reach out just personally with any questions after the talk. So thanks again. We really appreciate this talk. It was excellent.
Oh, it's a pleasure. Thank you so much for the invitation. Sorry, it was a bit long.
Thanks, everyone. Have a great day.